Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview authors from our feature articles, the papers of the month, or other articles in that month's print issue of the Diabetes Journal. We also have a Sweet Talk segment, which is a little bit more of a creative take on what's going on in diabetes research at the time. So my name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kevin Williams and Dr. Kirk Haberker. So nice to see you guys on this nice spring day. How are y'all doing?
Hey, Dar. I think we're in good spirits.
Yeah,
doing well.
Yeah, we have Kirk. We have an honored guest with us today.
Absolutely. We get to celebrate one of our own.
Yeah.
So-
Kevin not only was promoted this year, but recently was notified that he won the Nobel Prize for Creativity by the Society for the Study of Ingestive Behavior. Congratulations, Kevin.
Yeah, thank you.
And that comes on the heels of the Xavier Pai Sunyer
That's
Award
right,
that was
that's
earlier, right?
Yeah,
That's
that was
right.
in the
Yeah.
fall. Yeah.
At
So
the
this
Obesity
has been a
Society.
year.
Yeah.
Yeah,
It's all downhill.
Yep.
So
I think they got
congratulations.
me confused with some other Kevin Williams.
There are a lot of Kevin Williams.
Yeah, there
Did they
are.
put the W in there? Or what is your middle initial? Is it W?
Yeah, W.
Yeah.
Yep. That's a good question. I need to go back and look at that. They did put my photo up, so must be real.
Okay.
That's right.
Yeah, thank you.
I noticed I was looking at the award on the website, and Kirk, he gets money. So I think maybe drinks will be in free drinks or drinks on Kevin at ADA this year.
Yeah, the rule is the
Yeah,
money doesn't
absolutely.
leave the town, right?
That's
Isn't that the...
Yeah. We'll get you a couple hand grenades and hurricanes or something.
Oh. Do we return the cups for the refund? Is that right?
I guess if you want, you can take it as a souvenir.
take it
It
home.
depends on if you remember, like if you're still cognizant enough to hang on to the cup.
I'm sorry, but those hurricanes are a bit much for me. It's
They are.
basically high fructose corn syrup with a lot of alcohol.
That's right. Don't forget the dyes too.
Yeah,
I mean,
that's
they are
right.
pretty.
Yeah.
That's right.
All right. Well, congratulations, Kevin. And I guess we should focus on what's going on in today's episode today. So, Kirk, we have a type 1 diabetes themed discussion. And we should actually in advance apologize to the listeners because we had a little bit of a scheduling miscommunication between the three of us. And so I ended up soloing one solo interview on one of these interviews. So apologize to the listeners for that, but tell us about what we did for this episode.
That's right. So we're sticking with the beta cell this month, and the first article we talk about is IL-1 restricts progenitor programs and promotes beta cell maturation, revealing sex differences in diabetes progression. For that article, we are joined by first author Valeria Fabriciova, who is a PhD student in the group of senior author, Dr. Gabriela Pavlinkova. And then we discuss distinct enterovirus antigen landscape in children with islet autoimmunity with first author, Beatriz Delgado-Corrales, who is a postdoctoral fellow in the group of co-corresponding author, Dr. Ki Wook Kim.
Awesome. It's always fun. I learned so much with these interviews, and that islet autoimmunity one was definitely one that I learned a lot from. So yeah, this is great. Kevin,
Well, don't get used to doing them alone.
Yeah, exactly. And Kevin, we got to interview one of our friends and colleagues for the Sweet Talk segment. Tell us about that.
That's right. I mean, Kirk said that y'all kept it to the beta cell. We brought the brain into the Sweet Talk segment.
We did.
Our guest is Professor Lora Heisler, who was the recipient of the 2018 American Diabetes Association's Outstanding Scientific Achievement Award. She's also chair in human nutrition and head of the Obesity and Food Choice Division of the Rowett Institute at the University of Aberdeen in Scotland. And her work focuses on how brain circuits regulate appetite, body weight, blood glucose control, and she has really been at the forefront of how understanding these systems, these circuits, how understanding those circuits and how they behave have helped open new paths towards obesity and diabetes therapies. So I think it's a very interesting discussion about the past and the future direction of the field. So stick around to find out more.
All right, let's dive in. Today, we will be discussing one of the April issue's featured papers titled ISL-1 Restricts Progenitor Programs and Promotes Beta Cell Maturation, Revealing Sex Differences in Diabetes Progression. To discuss this paper, we are joined by first author Valeria Fabriciova, who is a PhD student in the group of senior author Dr. Gabriela Pavlinkova, PhD and Doctor of Scientists. She is laboratory head at the Institute of Biotechnology at the Czech Academy of Sciences in Czechia. Welcome, Gabriela and Valeria. Thank you so much for sharing your work with us today.
So, Gabriela and Valeria, we usually start with an icebreaker question. And so I'd like to hear from each of you. And I'm going to start with Valeria. If you could acquire any new skill in the world, what would it be?
Oh, skill or like do you mean like a superpower or just
Yes,
any
or
kind of
something you've always
skill?
wanted to learn, or, the floor is yours.
Oh well, I always wanted to learn play any kind of instrument because like I'm idiot playing anything and yeah, but otherwise it would be I would really love to be microscopic and get on the level of cell
Oh, yeah.
and this ultrastructure and maybe like on the molecular level to see how these machines are working. It would be fantastic, I think.
Yeah, that would be super cool.
Those are great ones.
Yeah.
Gabriela?
For me, I guess to learn somehow to get skills to learn languages easily, because that's always, I feel like this is really the issue. We have this proverb that we say, more languages you know, more times you can be person. So this would be my dream, that I could learn more languages than just Czech and English. I'm glad at least I speak English. So that would be thing I really would like to learn and do.
I love that. I think the more languages you know, the more people you could communicate with, and maybe our world would be in a better place right now.
Well
I
said.
would say so. Yes,
Yeah.
breaking boundaries, that would be my things. Yeah.
Yeah. Gabriela, we'll start with you. Are there any other authors or collaborators that you'd like to acknowledge for completion of this work before we talk about the nitty-gritty science?
Sure. Of course, the research is very complex, and I have to say it was a teamwork. But I would sort of split our group. I would name Valeria was really key person for single cell analysis. She has done all that single cell bioinformatics, beautiful pictures, and all the analysis. So that's, I want to stress this out. And another person would be Romana Bohuslova and Laura Lebron-Mora. They are also from my group, and they were really essential for the epigenetic analysis, for cut and tag sequencing. And Laura was mainly the bioinformatician behind this, and that was really, it was extreme work to do that, because it's I would say it's sort of new technology. So it was really, she was putting down the first analysis ever for our paper. And then group František Saudek, Zuzana Berkova, they are from actually hospital. It's called Institute of Clinical and Experimental Medicine. So they work on transplantation of human islets. And for us, what was important, their knowledge or their skills was all this quality control and in vitro tests of cells we were having on our hands. And I also want to name Klaus Kessner from UPenn. He is this pioneer person he done the first analysis on islet 1, on the transcription factor islet 1 in the pancreas. So, and we had really good discussion where we are going with our paper and what is this about. So I definitely need to name this person.
Excellent.
So that for this team.
And give me a little overview of your research program in general.
Our interests are transcription factors, how their dysfunction or their loss affects embryonal development. And we are looking at two different systems. The ones are neurons, and the second are endocrine pancreatic cells. And for the neurons, I'm developmental biologist, so sometimes it's difficult to delete transcription factor and see what is going on in the system, because it could be detrimental for the embryo, or you just cannot see it because it can be compensated. But we use the inner ear neuronal development. This system is itself isolated, and you can really follow the neuronal development, and you don't kill embryo or you don't kill the mouse. They just cannot hear, for example. And just starting from that, we developed our research, and we started looking also at the endocrine pancreas. Those are these cells are a little bit modified neurons, if you think about this.
Interesting.
They have a lot of things common.
Yeah.
Transcription factor networks. You look at the neurogenin, and it's in neurons, and it is in these cells to start with. So this is why this is sort of connected, and why we are looking at these both systems. So, and this is our, I would say, like main umbrella, looking at the transcription factors, embryonic development, transcriptomes, and now newly really looking. We were lucky to somebody created the cut and tuck, the way that we can look at the epigenome in a very small number of cells. This is really a breakthrough also in the methodology, to me at least, because for the embryos you have so many, you have such a small number of cells to work with. You cannot go to ChIP-seq and have millions of cells to analyze this. So this is really something which is moving developmental biology and the embryonal research really forward, and especially the epigenetic modification, because I think they were overlooked just because it is difficult to study them. They are important, but how you can find them and how you can study those. So
excellent.
All right, Valeria, we want to now move to you. And this manuscript focuses on the regulation of pancreatic islet differentiation. Can you tell us a little bit about why you're studying islet differentiation? Why is it so important? And can you maybe introduce us to the transcription factor that you focus on here?
Yeah, sure. So I think differentiation process is really important to study because it has major implications for diabetes research and for regenerative medicine. One of the key goals in this field is to generate fully functional insulin-producing beta cells or beta-like cells, to understand how endocrine cells are generated during the embryogenesis would provide us with better design and better protocols for how to generate these cells from stem cells. Another thing is really more like a new approach is how to regenerate these cells, like beta cells in situ, so right in the pancreas, in vivo. And I think to understand how transcription factors or chromatin modifiers are maintaining the chromatin landscape, or how they are keeping the maturity state, is really important for this approach. In type 2 diabetes, especially, there is known that these cells, these beta cells, they differentiate. So if we would find which factors are keeping them in this differentiated state, we would be able to find new targets for the more personalized medicine and like more targeted, like better targets for the development of new treatment. So yeah, and in my focus is islet-1 transcription factor, that is limb homeodomain transcription factor that is expressed widely in all kinds of tissues and organs. And the highest expression is during the development. And it is really important for the development of heart, inner ear, retina, and for example, motor neurons. And in pancreas, it is expressed in endocrine progenitors. Then during the adulthood, these expression levels go down, but still the protein is present and it is maintaining the mature beta cell state. Then islet-1 is also really important for the glucose-stimulated insulin secretion. I think it's a really interesting transcription factor because in our previous work, we showed that really it has an important role during the development of all endocrine lineages.
Awesome. So, Gabriela, tell us about you pretty much focus on one mouse model for this paper and use all these great techniques that you talked about in the introduction. Can you tell us a little bit about how this mouse model was developed?
Yeah, sure. So use conditional deletion of islet-1, and the reason is, as Valeria mentioned, it's absolutely crucial for the development of the heart. So if you do global deletion of this transcription factor, the embryo stops developing sort of in the first trimester for humans. In mice, it is really when the heart is formed into 4 chambers. So it's really important to have conditional deletions, otherwise you cannot follow later development. So we use NeuroD1 cream for deletion of islet-1 LOXP construct. So this is Cree-LOXP system, which allows conditional deletions. And actually, we created this model to look at the neurons. And then we found out this mice is also diabetic. So it was like a secondary target, I almost would say. But nevertheless, it's really very nice model because the NeuroD1 Cree is very specific. It deletes islet-1 only in the endocrine cells and fairly very early in the development. And it's also very powerful. So the deletion is very word successful or efficient would be better. The deletion is very efficient. So we are getting endocrine cells affected very early in the differentiation, actually almost at the beginning because the network comes, neurogenin is the first, then the NeuroD1 comes up, and after that is the islet-1. So it's almost like the third step in from the really early progenitors. So this is the model we have. And to describe, I don't know if Vale mentioned what we found first in the general phenotype in the pancreas was there are no alpha cells producing glucagon. This was when we were just looking at immunohistochemistry, looking, okay, why these mice are diabetic. So they have low total insulin. They are not able to have glucose-stimulated insulin secretion. So there is in vitro or in vivo, we cannot actually even inject these mice with glucose because they are not able. But however, even if you look at the phenotype, to me, what is really quite surprising is these mice can live up to 3 months of age. So they have no alpha cells producing glucagon. They have no somatostatin cells. This was just based on immunohistochemistry, and I'm sure Vali will tell you more what we found out thanks to the single cell RNA sequencing, when we could really look at each population individually and really see what's going on. So To us, this was really extreme phenotype. So the only cell which produces some hormone are the beta cells. So these are the only cells which differentiate into the stage that they produce insulin. So this is the only functional cell, endocrine cell left in this islet 1 knockout.
And in the title is sex differences. Were there any notable sex differences with these initial phenotypes?
In the initial, we noticed that the females are gaining weight normally, whereas males were affected much more. I mean, they were really behind their control little mates. Let's say in 3 weeks, we already could see the size difference in the weight difference. So that was something which we were discussing what's going on there, and that was more was then found out when we look at the single cell RNA-seq. But I think this should be question for Valeria.
Yeah.
She is really. She was instrumental to figure out the sex differences in this mutant. And I think it's really important to think about this, because I think this is now sort of really new thing that people are finally looking at the sex differences in their phenotypes, because before it was overlooked, or we just did not want to have, two different groups, males and females. We usually stuck with the males because they don't have any hormones, sexual hormones to deal with, and you have it's easier to have the analysis done. So, but now I think this is really important to have this shown and discussed. I mean, we have really concrete data showing that these sex differences are really early on, not when we have sexually mature mice.
As the token male on this call, I can confirm we are the simple version of the species. All right, so Valeria, I want to jump to the single cell seq, and can you tell us what the data revealed about the role of IL-1 as a transcription factor and its role in these various endocrine cell populations?
Yeah, sure. So the first obvious difference was, as Gabi mentioned, we don't have in our mutants delta population or the PP population. They are completely missing. And then another striking difference was that the alpha cells, they are still present in the mutant, but they're completely altered. They're very different from the control alpha lineage. They do express glucagon on very low levels, but otherwise they are missing some very important transcription factors typical for alpha cells. On the other hand, they have even upregulated some other transcription factors that are markers of alpha lineage. And so this is what was the contrast from previous work when we had bulk RNA-seq, when we couldn't figure out if we still have the present alpha lineage. We thought we don't have it. So now we figure out, okay, there are cells that were committed to be alpha cells, but they're just aberrant, not differentiated well without islet 1. Then another difference was that we had their endocrine progenitors that were hanging still in these postnatal ages, and they were not supposed to be there at all. In general, there were upregulated endocrine progenitor markers over all population of mutant islet lineages, but the endocrine progenitors itself created cluster on its own. Then when we looked at the beta cells, they were the ones that were the most closest to the control cluster of beta cells. So the only huge difference was that these cells, beta cells, were less matured than the control beta cells. this was the main findouts from the single cell analysis. And in general, what we found was that differentially expressed genes between control and mutant showed us that mutant has mainly upregulated genes. So this was kind of like directing us to the point that islet 1 should be a repressor, and mainly the repressor of these endocrine pancreatic progenitor markers. So yeah, this was what we figured out.
So I have a quick follow-up question, and that is, I'll expose my ignorance. I often think of islet development as going through the alpha cell to the beta cell, right? And in
Yeah.
this case, were you surprised that it was the alpha cells that were arrested, and yet the beta cells sort of kind of still made it to a pseudo-functional state? Am I understanding that correctly?
Well, beta cells, yeah, they were the only hormone producing cell, as Gabby mentioned, but still, it is really an enigma for us, like how this happened. That, yeah, at the beginning, when we looked at the earlier stages on immunohistochemistry, we found out that, yeah, up till embryonal day 13.5, we have alpha cells, like these primary alpha cells that are expressing glucagon, but then later they have diminished glucagon expression. It goes down, and
Okay.
we still don't understand how this happens, like what is behind this mechanism. So yeah, as you're saying, first we do have like the alpha cells, these primary alpha cells, and then they are able then to go to the beta lineage, but somehow the secondary alpha cells that are being generated like around the age 14, 15.5 of embryonal development, then there is a struggle. There something happens what is completely making them like aberrant and not very well differentiated.
Got it. Thank you for the clarification. So I guess I now want to talk a little bit about this pseudotime analysis. Can you describe what is the pseudotime analysis, and what did this analysis of that RNA-seq data reveal to you?
Yeah, so the pseudotime is a computational method that is placing single cells in pseudotemporal trajectory based on their transcriptome. So for example, even if we have just one age, we have postnatal day 9, and we want to see like how these cells are in their relationships one to each other. We will perform this analysis, and we will put them in this pseudotime, like from the initial point to the some ending differentiated point. So if I know that, for example, my cells start differentiation at, for example, cluster that is expressing highly some progenitor marker like FEF or NUROC3, I can place that as an initial point in the pseudotime. And then this algorithm will somehow place other clusters, other cells, to how they are related to this initial point. And I will see how distant they are. And I can say that, okay, these are more mature and these are more differentiated. And yeah, this is how it works. And what I found out in our dataset was that, for example, the beta cells of islet 1 knockout were really close to the endocrine progenitors that we had in the mutant. So what was it showing? That endocrine progenitors were mostly committed to become the beta cell, but also at the same time, the control beta cells were quite close on the other pole to these mutant beta cells. So the beta cells was the only lineage that was really kind of like really the most far away in the differentiation process, but they were not mature. And on the other hand, yeah, the alpha cells, they were like a little bit further away from endocrine progenitor population, and the control alpha cells were really far away from them. So it shows us like, yeah, these aberrant alpha cells are completely somewhere else in the differentiation process, and they are not very close to the control alpha cells.
Got it. Thanks. That was great.
So, Valeria, you next did a deep dive into the role of islet 1 in epigenetic programming. And I know there's a lot of data there, and I don't want to underscore the importance, but can you kind of summarize the critical findings from that work?
Yeah, sure. So we utilized cut and tag method for the analyzing the two histone modifications. We look at the H3K27 free methylation, which is the silencing mark or the mark of the closed chromatin, and then H3K27 acetylation, which is the mark of open or active chromatin. And we done this analysis for the mutants and controls. And my colleague Laura done the differential analysis of these marks. And what we noticed was striking difference between mutant and control, that the mutant cells had disproportional amount of acetylation mark, which is activating. So the chromatin was more open in the islet 1 knockout islets, and there was very little amount of the silencing mark. This was correlating really nicely with the transcriptome analysis when we found out that the majority of the genes were upregulated in the mutant. And when we done the analysis of looking at the overlay of these differentially activated genes and also islet 1 targets, we found out that, yeah, some of these endocrine progenitor markers there were there. And this is like what I found really interesting is that it looks like that in the differentiation process, it is like with the romantic relationships. If you want to progress to do and move on to the new one, you need to close the previous chapter, and islet 1 is providing that closure. And if you don't have it, it's gonna be mess.
Very nice. Very nice analogy.
So, on the topic of sex differences, maybe you can tell us a bit about what you found in your female islet 1 knockouts as compared to the male islet 1 knockouts.
Yeah, we first noticed this huge sex difference on 1 month old transcriptome. We noticed that, yeah, the females and males were creating separate clusters or almost like mirroring each other. But for example, there were different pathways that were specific for each sex. And we noticed that there were different antioxidative enzymes in females and males. There were different transcription factors and chromatin modifiers. And what we found out really interesting was that, for example, females had upregulation of long non-coding RNAs that play some role in the glucose homeostasis or in the circadian rhythm genes, which we don't know what is exactly the function. So I think it would be really interesting to study. And there was upregulation in the females in these estrogen responsive genes. So we think that the estrogen is the culprit behind some of these regulatory mechanisms, that maybe the females are better at handling the stress in the diabetes. And as we know, the estrogen itself is acting like antioxidant. There can be these kind of mechanisms that the estrogen responsive genes are better at activating some antioxidative or like stress responsive pathways. Another mechanism which could be behind these sex differences, I think, is, for example, different genome binding sites of transcription factors between sexes. Because, for example, we have upregulation of retinoid X receptor alpha in females. And it was already shown that in the hepatocytes, this transcription factor has different binding sites between females and males. So when we saw these huge differences in 1-month-old mice, we went back to the P9, the 9 days old mice, and we saw that actually, yeah, even this early, we have the sex difference in beta cells, where female beta cells are a little bit more mature than male cells. And we confirmed this on protein level, but it was very slight difference between males and females. Still, it was really like decreased level of maturation compared to the controls, but there was a trend. And when we look at the males, we saw more proportion in the enterochromaffin-like cells, which is like serotonin producing transient state in beta cell development. I think it is really important and interesting to study these sex differences, because I think it's going to be some detrimental differences in the pathologies especially, and this could lead to really some better personalized medicine approaches.
Yeah, and lots of follow-up work for a PhD or postdoc student, postdoc
Yeah.
training. All right, Gabriela, are there any other key takeaways from this work that you'd like to leave us with, or maybe some hints to some future directions?
Well, for me, it's the epigenome, which because we've been working with transcription factor and transcriptomes for some time, but the epigenetic modification and changes were always difficult to find out. And with this new methodology, with this cut and tuck, it's really super great that we can look at. And my future direction would be really not only looking at the single cell RNA or transcriptome, but also look at the single cell epigenetic
Wow.
change.
Yeah, it'd be really cool.
And this is really something I think we are going to develop, or at least we are discussing this, how this can be done, because you can see how it was believed that these just alpha cells were not differentiated, not there. But using new methodology, the single cell RNA-seq, now voila, we see all this population which are there and how they look and what this means. And I mean, sort of the future direction, of course, we are working in mouse model, and the second step would be, okay, how is this in humans, and maybe human stem cells differentiation. And actually, this transcription factor, islet-1, is really important for human cells too. And we have collaboration with Christina Nostro. She's from the University of Toronto. She works on stem cell differentiation. And this is, we have hopefully, well, I know I can tell you the paper has been accepted. So
Oh, yay.
it's going to be out very soon also. So there we show the islet-1 effect also in these cells. And it is same thing what we see. They saw our data in the mouse model, and they said, okay, it looks like we are getting sort of similar things. So then we start talking and comparing the data, and really this is what it is. The islet-1 has similar regulation as what we see in the mouse model. For example, these enterochromaffin cells produce extra, which is in human stem cell differentiation. You don't want to have this. This is contamination in their protocol, based on their protocols. So this is something which is really important to tackle. So I see always it's important to work in vivo. In mouse model is perfect because you can generate really great data which are really close to human endocrine pancreas. But it's also important to confirm and to have validation and somehow the connection to people. and the medicine.
Absolutely. Absolutely. Well, Valeria and Gabriela, I want to thank you for joining us today and sharing your expertise on pancreatic cell differentiation and the role of islet 1 in that process. I encourage listeners to read the full article in the April issue of Diabetes. Today, we will be discussing one of April's featured papers titled Distinct Enterovirus Antigen Landscape in Children with Islet Autoimmunity. We are joined by first author and postdoctoral fellow, Beatriz Delgado-Corrales, who is in the lab of Dr. Ki Wook Kim. Dr. Kim is a senior lecturer and principal investigator at the University of New South Wales in Sydney, Australia. Welcome, Ki Wook and Bea, and thank you for joining us today.
Thank you very much.
Thanks for having us. Yeah.
All right. So we can always tell whether you all are podcast listeners based on how stumped you are with our icebreaker questions, because we don't provide those in advance. But I'll ask you guys, how does your personal life intersect with your work? I'll start with you, Ki Wook.
Personal life intersect with. I don't know. I mean, my dad runs a cafe still, so he's been doing that for 20-something years, and so I often make myself a coffee on the way to work. So that's sort of how I but also I guess his sort of work ethics kind of did blend in through working as a team, working when someone's sick, filling in. So like that kind of did blend through my sort of scientific career. So he's actually a pharmacist by training. So it's when he migrated to New Zealand and Australia, he shifted his career path, and I also kind of did. I mean, I stayed in science, and Bear also has stayed in science, but we all shift a lot. So I did my PhD in plant molecular biology, and now I'm here doing human viruses and autoimmunity. So it's you never know where the life is going to take you. So yeah, that's enough for me. So
Bea, how about you?
It's similar. I'm similar to Ki Wook in the sense that there's not much that kind of intersects with it. I don't know why I chose to research in, type 1 diabetes, autoimmunity. I would say that I'm just, the reason I'm in Australia is 100% because of this job. So I think that's the part that intersects.
Yeah.
But I think I'm a bit of like a person that can go do research in different fields. And I chose this, I think, just because I wanted to prove myself that I could enjoy research in the medical field, because I tend to be a person that was like, I'm not touching that. I was used to doing environmental microbiology as a bioinformatician, which is another thing that, curiously enough, I always refused to be a tech person. Like everyone in my family and my friends, they remember me as this person that would not touch technology. And I just basically was like, I want to prove myself and everyone else that I can understand what code is. And here I am. But besides
there was quite a bit of coding and analysis in this paper that you're going to have to explain to us.
Well, it's just basically I'm doing this as opposed to everything, just to basically go against what everyone knows about me a bit. I'm like, you know what? I'm actually enjoying doing medical research. I'm actually enjoying doing bioinformatics, or just being more a properly nerdy researcher. But there's not much that intersect. I don't know why I'm so passionate about immunity in general, but it's just it came to be.
You found it and you love it, and that's all that matters.
Exactly.
All right. Well, Ki Wook, we'll start with you. Are there any other authors or collaborators that you'd like to acknowledge for the completion of this work?
Yeah, I mean, we have co-authored Professor Heikki Vuoti from Finland, so he's been a long-term collaborator with us, and he's also kind of been we deem him as sort of a father figure in the viral etiology of type 1 diabetes field. So we really acknowledge his intellectual input and feedback from early in the conceptualization of this work. And I guess it may relate more to the next work that we are doing as a follow-up of this, but I'd like to acknowledge Professor Stephen Alledge from Harvard, who sent us the VerScan library. So we've been doing further analyses using that phage library, and our active collaborator in UCSF, which is Professor Joseph DeRisi. Yeah, so he's also a very well-known figure in the field of virology, and he's been really instrumental in helping us progress this work further. Yeah.
That's great. So, can you give us an overview of your research program and then kind of elaborate on how it dove into this particular project?
Sure. I mean, it stems back to his co-joint senior author with me is Maria, who's a pediatric endocrinologist, and she's also an active researcher in this viral etiology field. And it's really decades or almost a century of epidemiological data has associated viruses with type 1 diabetes. And that observation led to really a lot of work done over the few decades, especially in the last few. And Maria and I, we've put out several papers and systematic reviews that have really shown a very strong association between individuals who develop autoimmunity and type 1 diabetes having some form of enterovirus in their system, whether it's viral nucleic acid or protein. So we've been really following this path. But one of the problems and criticisms has been the fact that it may have been too targeted. There's been decades of work done on enteroviruses, but what about all the other viruses? When I first started working with Maria, which was about 8 years ago, we started to think about how to explore the virome as a whole, as a holistic approach, and looked at different technologies that can do that. So we've been focusing on trying to come at it from an unbiased way and then kind of zoom in on whatever signals that we find. So that's been our approach, and what we've been able to really focus in on this work and the last few years has really been which viruses do find an association with the development of autoimmunity, especially in islet autoimmunity in young children. So we have a nationwide prospective cohort called ENDIA. It stands for Environmental Determinants of Islet Autoimmunity. And that cohort's been pre-existing from before my time in type 1 diabetes field. So 2013, the earliest child was recruited, and now some of the children have already age 10 or past that. But really the power has been to prospectively sample from pregnancy and then early childhood and see whether there is sort of a environmental trigger that occurs and precedes the development of the first islet autoantibodies. And that's where our focus has been, to look at that time point, look at what viruses are there, look at it from different angles, whether it's nucleic acid or in this case, it was antiviral antibodies, and trying to explore, what we see, and do we see what everyone else has been seeing with the enteroviruses. Yeah.
And so Ki Wook mentioned one of them, but you used two prospective birth cohorts. So can you tell us a little bit more about those cohorts and then sort of the subjects that you ended up utilizing for this particular study?
Yes, of course. So apart from India that Ki Wook just described, we've also used another Australian cohort called the Australian Viruses in the Genetically at Risk, or VIGOR. That's what we call it. And basically, we wanted to use these two Australian cohorts as a matter of not only investigating, but comparison between two kind of, they're similar in some ways, but also different in others, which makes it like really interesting to compare. So I'm going to start with VIGOR because it's the oldest cohort that we have. And it's basically a cohort of children that were selected based on having one or more first-degree relatives with type 1 diabetes, pretty similar to India. Then the samples were collected for these children between 2006 and 2015 at time of islet autoimmunity onset. That's the samples that we're actually analyzing. It's a small cohort. We only have 21 individuals, 21 individual cases and 21 individual controls. So like a 1-to-1 ratio. But basically, it's just the oldest cohort that we had, and also with children that range over 1-year-old until 12 years old, which is fairly different from India, because even though the India children were also selected based on having a relative with type 1 diabetes, could be mother, father, sibling, but they were recruited and the samples were collected between 2013 and 2018. Also, both have in common that this was pre-COVID, which is very interesting.
Yeah.
Yeah. These children, the India children, are really young. They were also selected, the samples were selected at islet autoimmunity onset, but they were really young. Like the children, I don't think most of them are less than 4 years old.
Okay.
The ones that we've analyzed at least. Now, there's more children that are seroconverting and moving to type 1 diabetes, but those would be for other analysis in the future. To summarize it a bit, I think India is younger children, also a 1-to-3 ratio. So we had three controls per case. Cannot remember exactly the number of samples that we had. I think it was around 42 cases, and then more or less 94, 93 controls. Cannot remember exactly, but we try to keep the 1-to-3 ratio. It's just because of sample issues, or just basically that the nests at the time were not fully complete. We might have 1-to-2 ratios versus 1-to-3. But overall, that's kind of the setting. Samples for both cohorts were taken at onset. And and also Vigor is a much smaller cohort. I just want to stress that, because in terms of power analysis, you will see that India is more powerful as a cohort, especially because we are analyzing a nested case control study versus a more not nested case control study in Vigor. But that's kind of a summary of the two cohorts.
Okay. And then what endpoints were you looking at?
Generally, what we wanted to see, we wanted to identify the overall viral antibody profiles of cases versus controls in both cohorts. Again, when I'm referring to cases, I'm referring to children that are islet autoimmunity positive, that have seroconverted. We just wanted to see an overview, and in an unbiased way. That's the power of the analysis that we're doing, VERSCAN, which is basically you just have a look at all the antibodies available in the plasma samples. And not only we are able to know what viruses they're targeting, these antibodies that we find in these plasma samples, but we know what area of those of the viral proteins they target, which is really good. Like you have something that's more broad, that we can have a look at just basic antiviral antibody differences. But also we wanted to focus on, can we see a specific targeting of viral species? Can we see what antigenic regions are being targeted? Is there a difference between cases and controls? If there is that difference also observed in both cohorts or not? Is it conserved across cohorts? That was kind of our idea. But as Ki Wook said, we tried to approach this project in a more unbiased way, to have a look at overall antiviral antibody profiles. And then, of course, we know enteroviruses have been associated for decades to the onset of islet autoimmunity and to type 1 diabetes. So we also wanted to have a particular look at enteroviruses too. But we started with this very unbiased kind of goal.
In your initial analysis, you saw some differences in the frequency of the enterovirus and islet antibody in the two cohorts. So talk a little bit about that.
So what we initially saw was that for both cohorts, enteroviruses were kind of the main players there. Antiviral antibodies that we found were targeting other viruses. I want to say species, but I'm just going to try to say just genus in general, because species is a tricky question, as you'll see later. But we saw a wide range. But in something that both cohorts had in common was that antiviral antibodies against enteroviruses were extremely common and extremely high in counts. Let's say it like that, because we do have, we work with DNA sequencing data, so we call it counts. So we did see that. However, when we start to have a look at, are there any significant differences between antiviral antibody counts or even composition between cases and controls in these cohorts? Do we find any? And we couldn't say so. It's kind of like that overall, both like children that are cases and children that are controls do not have that much difference between them in terms of overall antibody levels against any other virus, and specifically against any enteroviruses. So is it surprising? I think not really, because enteroviruses are so common. I cannot stress enough how common they are at really young ages. Finding that cases have, imagine this situation that cases do have enterovirus antibodies but not controls, that would be really surprising, because that means controls are not exposed to enteroviruses, which again, not really that common. So at first, that's what we found. We didn't really find any significant differences. It was more about how common enteroviruses were, and that's what we had to kind of change a bit, what we were trying to search for later on.
And then you took a deeper dive into the enterovirus specific immune responses. And do you want to try to touch a little bit on the analyses as well, just to kind of describe it for our listeners?
Yes, of course. We based our studies on a previous publication, if I remember correctly, by the Joe DeRisis lab, where they did something that we like to call antigen mapping. So because we couldn't find an overall difference between cases and controls just in the number of antibodies that were targeting enteroviruses, we decided to have a look at, okay, there's no difference in the overall, but what if we actually have a look at the specific antigenic regions that these antibodies are targeting? Is there a difference? So are they targeting different regions, for example? Are the levels of targeting different? And that's what we basically hit the jackpot. So to kind of explain it in a simple way for the listeners, what we did was we used a full enterovirus genome in its protein version as a reference. So we basically put it out there to have a look at where our antibodies were targeting. We used an alignment method to take the sequences that we knew the antibodies were targeting, that basically VerScan can tell us that, and we align them to this reference genome over and over and see where these regions were laying across the genome. It's interesting because we find that the main regions that were being targeted in general for both cohorts were the VP1 region, which is an external protein, which are in our minds, and especially I'm a microbiologist too, I will always assume that that's the main antibody targeting region for a virus, the capsid proteins. But we also find really interestingly that a lot of the antibodies were targeting the 3D region, which is a region that encodes for the RDRP, so the RNA-dependent RNA polymerase. That's a very conserved protein, but also when you have the assembled virus, it's not there. It's only produced after the virus enters the cells and starts transcribing and starts translating its proteins. So that was really interesting. That's basically what we did and what we call antigen mapping. And you can do that with any virus, actually. If you have a reference protein or a reference genome in its protein version, you can do that with any virus. It's just enteroviruses are kind of, we were more interested in this because we knew already by the literature, by our own experience, that there was some signal that
there was some connection. Yeah,
exactly. So that's basically what we did, and that's where we found what we found. And it's that basically once you have the antigen map, you can detect what regions have been targeted. And again, there are no differences in targeting in what regions are being targeted between cases and controls. So both cases and controls targeted the VP1, both cases and controls targeted the 3D, but the levels are radically different. The when you look at the increase or the differences in increase between cases and controls on where they target the VP1 and the 3D, cases target so much more the 3D. The level of antibodies targeting the 3D or RDRP, whatever you want to call it, is significantly different between cases and controls at onset time.
Yeah.
With the VP1, the signal was a bit more confuzzled, I like to say. So there was not a clear region that was more targeted. There was something going on there, but there was no pattern that was clear to us. But with the 3D, it's the fact that it was not only significant, but conserved across the two cohorts that had nothing to do with each other.
Yeah.
Were recruited at different times, processed in a different manner, and you
Yeah.
can still see that signal there. That was really interesting, fascinating.
Yeah, suggesting that the signal is more robust.
Exactly.
Right.
And that it is just conserved. Yeah.
Yeah. And then you saw some sex differences that kind of elucidated a little more about the system or interaction, correct?
Yeah. So when did it come? I think it was me and my co-author, Dr. Legana Fingerhurt, we basically loved, after we got the antigen maps nailed and we got them right, we're like, okay, let's just stratify this by all sorts of things. We try even HLA, but the more you stratify, the less, like, the number of samples just keeps being reduced and reduced.
Yeah.
So there's just so much that we could do. But we said, we know that the immune system in females and males is different. We know this especially in autoimmunity. Like females, the rates of autoimmunity in females is just off the charts compared to males. So we knew this, and we're like, let's just stratify by sex and see what happens. And voila, then you have the VP1 region being targeted. Yes, the targeting was conserved between the two cohorts, but then you see that females and males have completely different targeting. Something that was present in males, actually significantly present and enriched in males in the VP1, is not present in females, which is, what does this mean?
Yeah.
What does this mean? And that's just a hint of something that we need to explore more. Because for now, I mean, again, why are the immune system reacting different in females and males? People might have a lot of theories. The X chromosome might play a role there, but it's something that we need to explore. But this might be, I don't know, could be associated with why islet autoimmunity presents differently in females versus males.
Yeah.
It might be, but the fascinating part was that the 3D, even when you stratify by sex, the 3D enrichment was conserved still across males and females and across the two cohorts. But then also that the VP1 now has a consistent pattern that we can trace in both cohorts, and there's a difference between females and males, which is, again, it was just really interesting.
Yeah, that's really great. Ki Wook, are there any other key takeaways from this work that you'd like to leave us with, or maybe give us a hint on some of the future questions that you or the field should address?
Yeah, thanks for this question. I guess we feel that there is a clear signal that is very distinct in the case children. And as Bea mentioned, these children in India particularly, they're very young. So they peak at islet autoimmunity around 2 years of age. We actually think that rather than the past field, the past few decades have really focused on which type of enterovirus, which serotype, which species of enterovirus are what we call diabetogenic, so they cause or trigger some event that elicits type 1 diabetes. We actually kind of challenge that. I think our data really supports that it's maybe not so much the type of enterovirus, because when you actually have them at amino acid level, so what they present to the immune response, These RNA viruses, they're very, very conserved in these regions, VP1, 3D. They're very, I would say they're looking very similar to each other. So how would one type elicit something very unique that doesn't occur in controlled children? I think it's a lot more plausible to think that we are all the children are exposed to these ubiquitous enteroviruses, and it's just something about them, their host response, and that may be driven by genetic risk or other factors. How they respond is what's really causing this islet autoimmunity to be triggered. I think that's a lot more plausible. And if we take that kind of model, then I think our data really fits where we have this very distinct signal from the 3D region. And I think whether we find that this really is pathogenically relevant, or if it isn't, at least we actually think it might be a much more early biomarker compared to what we normally detect and monitor for islet autoimmunity. So to actually diagnose or deem someone islet autoimmunity positive, there are 4 classical autoantibody markers that you serologically test. But these peak around year 2, whereas enterovirus infection can occur within the first year of life or even sooner. So we actually think there might be a value in terms of an early biomarker even, and that can lead to potentially better strategies for earlier intervention, enrollment into clinical trials. So I think that's one key relevance that we are pursuing at the moment. But we also have our theory about, we actually think this is pathogenically relevant, and we are actually trying to think, and we tried to hint this in the paper, that delve deeper into what this motif in the 3D region actually looks like, and whether there could be any potential molecular mimicry happening. And if that's the case, I think that would be really remarkable if we can find, and there's been, not with type 1 diabetes, but other autoimmune conditions that have been shown that sites of viral antigens can actually be key places for molecular mimicry. So we're really pursuing those 2 areas, and we'd like to, be excited to share that with you in the near future.
Yeah, we look forward to it. Sounds really great. So, Beatriz and Ki Wook, we want to thank you for joining us today and sharing your expertise and discussing your recent work on the Enterovirus Antigen Landscape in children. I encourage the listeners to read the full article in the April issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, here with my co-host, Dr. Darleen Sandoval. We're delighted to have Dr. Lora Heisler joining us today. Dr. Heisler is professor at the University of Aberdeen in Scotland. Over the course of her career, Lora's work has helped define how brain circuits influence appetite, body weight, physical activity, and blood glucose control. She's also the recipient of the 2018 American Diabetes Association's Outstanding Scientific Achievement Award. In recognizing her, the ADA highlighted her seminal work that clarified how serotonin pathways could be harnessed in the treatment of obesity and type 2 diabetes. Lora, it's a real pleasure to have you here today. Thank you so much for joining us.
Oh, thank you so much. I'm really excited to talk to you guys.
Now, we usually start these things off with a little bit of a icebreaker question, and I was curious, what scientific question in metabolism or neuroscience still feels the most exciting or maybe mysterious to you?
Well, that's a really good question. I think what fascinates me is the brain's astonishing ability to calculate the calories that we consume with incredible precision. So before the modern food environment sort of disrupted things, it was a disruptor. Adults typically maintained stable body weight within a very small margin of error, about a pound a year. So I remember my first lectures that I was giving, I was talking about that, how people tend to only gain around a pound a year. And so from 25 to 55, that's 30 pounds, and that's the middle age spread. But if you actually think about the energy that's involved in that, that's equivalent to miscalculating each day by about a single potato chip or a sip of orange juice. And this is just like crazy, remarkable biological accuracy. So to me, still, the great mystery is how the brain performs this calculation, and importantly, how the modern diet overwhelms this finely tuned system. So understanding that disruption and how to restore energy balance remains one of the most exciting research questions to me.
Yeah, it's a really great point how tightly regulated that is, and trying to understand that mystery of how the brain and the body is tightly regulating energy balance is really interesting. Now, in the introduction, we highlighted that you were recognized with the Outstanding Scientific Achievement Award with work that helped identify how the field thinks about energy and glucose homeostasis. So I was curious if you could take us back to the beginning a bit. What first drew you towards a career or into research of metabolism, appetite, and the brain?
So you're right. It was it was quite some time ago, and I want to thank the ADA for really launching my independent career. I was very lucky to be the recipient of an ADA Junior Faculty Award that allowed me
Nice.
to pursue a very unconventional idea. So when I was a postdoc at Harvard Medical School around 2000 in Joel Elmquist's lab, I generated some preliminary data that suggested that the brain might directly influence peripheral insulin sensitivity and blood glucose. So what happened was I treated a mouse with a serotonergic drug because I was interested in studying brain circuits that control food intake and body weight. So I knew that this drug only acted in the brain, and what I found was an immediate improvement in blood glucose and insulin sensitivity. So there was no time for a change in body weight. It just happened, inject drug, immediate improvement in blood glucose and insulin sensitivity. And at that time, the idea that the brain could be involved in blood glucose or insulin sensitivity was considered pretty crazy. And my colleagues told me that it couldn't be right. So, just the colleagues from all over said, no, it doesn't make any sense. Diabetes treatments target peripheral organs, not the brain, and they work. So there's something going on here, but your idea that the brain could be involved can't be right. But I was just really lucky that the ADA took a chance on this unconventional idea and funded the research through this junior faculty award. And, fast forward 26 years later, it's now widely accepted that the brain plays a role in glucose regulation. So what once seemed like a far-fetched idea is now just, yeah, sure, it's dogma. This is just one of the ways that blood glucose is controlled.
Yeah, I think that's a great point to think back at the beginning of that, beginning of your career and the beginning of the field as it relates to the time that you're studying this, and how underappreciated the brain could have been in driving some of these improvements in blood glucose regulation, and even as it relates to body weight regulation, and how that has changed so much in just a couple decades. And that today it's nice to see an appreciation of what could be happening with neural regulation to drive changes in body weight and blood glucose control. And you were definitely a major driver of that, and the ADA recognized you for that work.
Yeah, it was fantastic to even though the average person at a conference or colleagues were like, I don't think so. What was really great about the ADA is that they said, try it, let's see if it's right. And it turned out to be right. So that was really gratifying. But as you said, like, initially what I was looking at or initially what I was investigating was how serotonin circuits influence food intake and energy balance and body weight. Initially, you would imagine that serotonin, because the majority of serotonin is made in the gut, it would make sense that that's where serotonin affects food intake. It's very intuitive to imagine that. But 50 years ago, researchers found that the relatively tiny pool of serotonin in the brain is sufficient to have profound effects on food intake and body weight. And so the challenge and the opportunity was to uncover where in the brain serotonin acts and how these circuits influence eating. And that's what I was studying. So I think one of my biggest career discoveries is figuring out a key mechanism of action of the previously widely prescribed obesity medication, defenfluramine. The way that it works is by boosting endogenous serotonin. So this work helped map those pathways and clarified serotonin's central role in energy balance.
Yeah, so 20, 30 years ago, the focus was on the melanocortin system, right? That was sort of, we were really picking apart that whole, and you ended up connecting serotonin to that system. Can you tell us a little bit about that work?
Yeah, that was interesting. So the focus of my PhD was on, can serotonin be exploited as a therapy for obesity treatment, and then that was defenfluramine. Then defenfluramine led to, because it was just increasing serotonin bioavailability everywhere, it was having off-target effects in the periphery. So one of the challenges became to dissociate the therapeutic benefit from these peripheral side effects. And that's when I turned my attention to the brain. Then sort of thinking about, well, what's produced in the brain that might be sort of a key mediator of appetite and body weight. And so, so just thinking again about serotonin. So since serotonin itself doesn't cross the blood-brain barrier, the options are in sort of drug development is to boost serotonin's own bioavailability, which is what defenfluramine did, and sibutramine, and then also develop serotonin receptor agonists. So once defenfluramine was demonstrated to produce side effects, then the next question was, well, which receptor is it acting through to produce most of these benefits, and what is that receptor mediating? And so that's where I was looking at the serotonin 2C receptor in my first postdoc, and then what the serotonin 2C receptor was mediating in my second postdoc. So my first postdoc was at UCSF with Larry Teacott, and my second postdoc was with Joel Elmquist at Beth Israel at Harvard Medical School. And as you say, what we found was that serotonin is influencing melanocortin pathways to produce its therapeutic benefit. And so how was that discovery made? It was really just thinking about, well, what are the primary mediators of appetite and body weight in the brain? And so it was thinking about, well, what are genetic forms of obesity? Because that tells you that's a fundamental circuit. So I worked with the AY mouse, which is a mouse that overexpresses agouti, and that stimulates hunger and body weight. And as you will remember, that what was worked out around that time is that there are sort of fullness promoting neurons called pro-opiomelanocortin, and they're competing for action with these hunger promoting neurons called agouti related protein acting at the melanocortin 4 receptor. And so what we identified is that serotonin is acting through that melanocortin circuit, that fundamental melanocortin circuit, to produce its therapeutic benefits. That's one of the primary ways that it's doing it.
One of the interesting things that kind of strikes me is your work sort of crosses that physiology and pharmacology kind of aspect of serotonin, right? And with GLP-1, the pharmacology is mimicking the physiology dogma, but the physiology probably isn't quite what everyone thinks it is. Did you feel like you kind of encountered that with serotonin as well?
That's a really good point. I think the difference there is that what was exciting about difenfluramine and sibutramine is that the only thing they were doing is boosting the body's own endogenous serotonin. So it wasn't pharmacology. It wasn't overwhelming the system with levels that the body has never seen. It was actually just tapping into what the body was already making. So that's a difference where I think the GLP-1 receptors, the GLP-1 receptor agonists are producing a higher level of action than what the endogenous GLP-1 would be doing. But you, of course, this is your area of research.
Sorry, I had to
So
sneak GLP-1 in there.
No, I know. I'm excited. You have to. It's super exciting. But I mean, you know better than anyone about the pharmacology. But I think that's just a really good point. Is that the GLP-1 is much more pharmacology, whereas serotonin does have that sort of advantage of being also physiology.
Yeah.
But I will say that the incretin therapies are transforming obesity treatment, and it could be that the degree of weight loss achieved is because of the pharmacological action, not sort of a physiological response but a pharmacological response. But I'm sure you remember back in the day, I'm sure that that I think it's just worth pointing out that GLP-1 receptor agonists effects on body weight. This is not new information. I remember in the early 2000s, people arguing about GLP-1 receptor drugs, in these heated debates at conferences. And some people saying, yeah, but GLP-1 receptor drugs only produce effects on food intake and body weight because they cause nausea, and therefore they have no therapeutic future. So what's new is not that the drugs do this. What's new is that patients are willing to tolerate these side effects because body weight is so difficult to shift. It's so challenging. And they're willing to accept the side effects in their weight loss journey because they're desperate to lose weight. So I think that is what was a big surprise to many people. I don't know, was it surprising to you?
Well, I mean, I think the dose escalation tries to avoid that, and then, but I think what's fascinating is the tachyphylaxis of the nausea, right? That it can go away over
Yeah.
time, and how the heck is that happening? I think that's really
Yeah,
interesting.
that's a really good point.
But it doesn't
as
for
well. But I
everybody,
suppose-
right? So some of those people that are really like they just can't tolerate it, and so they end up going off. Yeah, it is very interesting, but I think it just speaks to the enormity of the problem, right? And
Exactly. It does speak to that, and I think that's something that was not expected. It's quite interesting how a decade ago or 15 years ago, the sort of overall perception was that obesity is going to be incredibly difficult to treat with a medicine, because obesity is caused by a range of factors. There's not one thing that leads to obesity. There are many things that lead to obesity. So having a single drug then influence body weight seemed like it was going to be a really big ask. And so for
yeah,
that reason, a lot of drug companies just sort of mothballed their obesity programs and thought it's not, we're not really sure this is going to be able to be treated. But now there's this resurgence of interest in it, because of this recognition that actually it can be treated effectively with the GLP-1 with incretin therapies.
yeah.
Building on this, with therapeutic intervention, the development of drugs to treat obesity, type 2 diabetes, a lot of the work that you've been involved with from pretty much the inception was at the new age of molecular genetics that was being brought in to study obesity, food intake, appetitive behavior, blood glucose regulation, especially through these neural networks. And it's really an interesting use of tools to perform a mechanistic neuroscience approach to refine potential therapeutic strategies. And I'm curious if you were looking back at the serotonin story, or even as you're talking about now, the incretin story with treatment of obesity and diabetes. Do you think there's lessons that can be learned in those approaches of how basic science can be meaningful in shaping drug development?
Oh, definitely. Yeah, I think that is maybe what a lot of people aren't aware of is just the how exciting the tools that we have available as researchers are, which does allow us to make fundamental discoveries. So just, to give you an example, so, just along this effectively 26-year journey, our work along with others like yours, Kevin, and yours, Darleen, has shown that specific brain circuits play a central role in appetite, body weight regulation, and glycemic control. So we're working out this circuit that serotonin acts at the serotonin 2C receptors, influencing cells producing the endogenous melanocortin 4 receptor agonist. And just that pathway has led to 3 different therapies that have 3 different obesity medicines. So you had difenfluramine and sibutramine, which were augmenting serotonin, endogenous serotonin. You had lorcaserin that was acting at the serotonin 2C receptors. And now you have setmelanotide, which is acting at the melanocortin 4 receptor. So it's a melanocortin 4 receptor agonist. And then also looking at, well, how does GLP-1, how do GLP-1 receptors fit into this? Are they part of the same circuit, or is it multiple circuits? How are they producing their effects? So it is quite surprising that many of the sort of fundamental regulators of appetite are acting through this really primary circuit, including things like leptin is also acting through the melanocortin system, but elsewhere as well. So I think GLP-1 is, the GLP-1 receptor agonists are also working into this. But what's exciting is that today's tools allow us to interrogate the function of specific cell types within discrete, defined brain regions in ways that were completely unimaginable 20 years ago. So we're living in a transformative moment in neuroscience and metabolic research. And it is just like so exciting to be a part of it. I genuinely love it. And I'm sure you guys do too. It's just really amazing.
Yeah, it is really fun. And I mean, at least in the States, things are a little bit more challenging for training, but you've built a research program across multiple institutions and countries. So
Yes.
what do you try to instill in trainees entering metabolic research today, and especially thinking about the technology, the big data, AI? We have so many we're kind of on the forefront of some technological breakthroughs. So what are you thinking about for training young scientists now? Probably trying to
Yes.
recruit some
Well,
from the States.
I know, that's right. That's right. So I think for me, translation's always been the goal. So I've been extremely fortunate to work with extraordinary mentors like Joel Elmquist and Larry Teacott. And Kevin, you also worked with Joel. He's
Yep,
fantastic.
yep.
I mean, he's just a really, really fantastic mentor. And I've been lucky to work with talented colleagues and have really fantastic research teams as well, who all sort of share that translational mindset. So I would say for me, the best scientific questions have naturally led me into spaces where mechanism and clinical relevance intersect. And I think that's where the most meaningful progress happens. So back when I was doing my first postdoc, my supervisor, Larry Teacott, said, if the effect isn't big, it isn't worth chasing. And that's because the goal for me, the ultimate goal, is to translate to another species, to people. So we're looking for a discovery that is fundamental mammalian biology. And as we all know, time and funding are finite resources. There just isn't enough of either one of them. So to make a real impact, I think a good approach is to go after questions that yield robust and meaningful effects. And that can be quite hard if you're in the middle of a project and you really like it and you're sort of excited by it, but your hypothesis is not being supported. It's quite hard to just stop. But that's my advice: is if your hypothesis is not supported and there was nothing wrong with the methods, then go after something bigger. Or if the effect is small, go after something bigger. And that can seem quite difficult, but that would be my advice. So the other thing I would suggest for people to think about, really the big questions. If you don't ask a big question, it's going to be hard to make a big discovery. So if it's a small question, you'll get a small answer. So go for bold and risky projects where the big discoveries may lie, but then also keep going with safe and steady projects. So that mix will help maintain momentum while still leaving room for creativity and discovery. And also, if people are telling you, well, that can't possibly be right, if you've got preliminary data supporting something, have the courage to go after it, even if people who are more established than you are questioning it. I think still go after it, and if you get funding for it, definitely go for it. It's an incredibly exciting future, and I feel so privileged to contribute to the basic science side of this journey to help in whatever way that I can to shape what patient care might look like in the future.
Yeah, I love that, the big effect comment, because it is really hard when folks they are very enthusiastic about their hypothesis, right? But then they get the data and the effect is really small and you kind of have to squint to see it. And there's
Exactly. And that's the difference between serotonin in food intake, and GLP-1 receptors in food intake. When you give a serotonergic drug and you measure food intake, you get, again, it could go down to dosage, but you get around a 30% reduction. When you give a GLP-1 receptor agonist, you can get an 80% reduction. It's a big effect and it happens every time. And that has led to this translation. But I think for me, it has been about chasing going after the big effects and leaving behind things that just weren't big enough, because you need to see it every time in every lab and for it to have any chance in having it translate to people. But I think one of the things for me that was sort of the most gratifying was going with that idea of, why would the brain regulate peripheral glucose? It doesn't need to. It's seemingly unnecessary and counterintuitive. But believing that biology builds beautiful and complex systems. And, so something that might seem initially counterintuitive then might end up being something that just makes total sense. And it's just the way that you're looking at it. We know the brain controls homeostasis, so it makes sense. And the brain is ultimately the body's command center. So I think that's been a really exciting part of my career, is having an idea that sounded cuckoo and then finding that it works in patients. That was really gratifying. And and there's so much more to discover. I think that the tools that we have now are just phenomenal. And I do believe the next decades will bring a huge shift in understanding, and I can't wait for it. But I think there'll be a shift towards gene therapy and optogenetics and chemogenetics and other transformative approaches. And where we are now, the challenge and the opportunity is figuring out how to bring those technologies from cutting edge research that we're all doing in our laboratories into mainstream clinical treatment.
Lora, this has been really great, a really great chat. So many life lessons here, little nuggets. Before we go, could you let our listeners know where they might follow your work? Do you have a lab page, institutional page, social media presence? Where could you point them to?
Yeah, so I have LinkedIn, so it's just Lora Heisler, but you have to remember that my name is spelled in a funny name. It's Lora. And I also have a website again, so it's just University of Aberdeen. But I think if you type my name into Google, you should come up with the University of Aberdeen. And if you type my name into LinkedIn, you should be able to find me as well. And I've got a Blue Sky account that I'm just starting to use, but I'm not really. I'm not really on X or sort of Twitter anymore. So, although
Yeah.
I used to be active on it, I'm not really doing it anymore.
Thanks again for joining us. And to our listeners, as always, please like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.